So good evening, students. So let's begin this session, right? So in the previous session, we started off with the genetic disorders. So I gave you a brief introduction about these genetic disorders. And also we saw a few basic classification of how we actually start classifying these genetic disorders. So getting through a quick recap of what we have seen in the previous session, right? So what exactly is this genetic disorders all about? So if a particular disorder is caused because of a mutation in the gene, or a mutation in the chromosome, we started calling it as genetic disorders, right? Either there is a mutation in the gene or there is a mutation in the chromosome. So because of these two reasons, if a disorder is actually caused, we start calling it as genetic disorders, right? So we saw that the first genetic disorder was called as Alkaptonuria, which was first identified by a scientist by name Garrod, right? Sir Archibald Garrod in the year 1908, right? In 1908 or 1902, he identified this particular disorder. Then we saw a few things also with regards to Alkaptonuria, right? Now, and then along with that, we saw also the basic classification of how we actually classify all these genetic disorders. So all the genetic disorders could be classified broadly into two categories, right? One is gene disorders. The second one is called as chromosomal disorder. So when we talk about gene disorders, so what does that mean? A disorder which is actually caused because of a mutation in the gene present on a specific chromosome. We call it as gene disorder. The other side we took it up as chromosomal disorder. So what did we see in chromosomal disorder? If a mutation results in either change in the shape of the chromosome or increase in the number of chromosomes, that type of disorders we started calling it as chromosomal disorder. Then again, under gene disorder, we also saw called gene disorder as Mendelian disorder or inherited disorder, because those are the disorders which are actually inherited from generation to generation. We can also call it as inborn disorder. So a person, a child would be born with such sort of a disorder. Whereas chromosomal disorder, we call it as non-inheritable disorder because they are not inherited from generation to generation, right? One of the major reasons for that is because most of the individuals who are affected by this sort of chromosomal disorder will not be able to undergo the process of reproduction. Mostly they are said to be infertile, right? Or they are said to be sterile. That is one of the major reasons why we start calling it as uninherited disorder. Right. Now, again, with regards to gene disorder, we classified gene disorder into two categories, right? Based on which type of chromosome is getting mutated, or the gene present on which type of chromosome is getting mutated. Whether it is autosomal gene that is getting mutated, or whether it is X-linked genes that are getting mutated. Right. So when we say autosomal genes, what does that mean? Genes that are present on the autosomal chromosomes. So if there is some mutation in the genes present on the autosomes, right, such sort of a disorder, we call it as autosomal gene disorder. If the disorder is caused because of mutation in the gene that is present or situated on the X chromosome, we start calling it as X-linked gene disorder. Again, autosomal gene disorder we classified again into two categories. One is called dominant disorder and the other one is recessive disorder. So if a disorder is caused because of a dominant mutation, we call it as dominant disorder. If a disorder is caused because of a recessive mutation, we started calling it as a recessive disorder. Same case, we saw it even with regards to the X-linked disorders also, right? Either dominant disorder or a recessive condition, recessive disorder. Right. With regards to chromosomal disorder, again, we took two different types. One was called, again, the same thing, autosomal chromosomal disorder, and other one is allosomal chromosomal disorder. So in this session, let us take up a few disorders in each of the categories, and we'll see what exactly are the conditions seen or what sort of mutations is seen in all of those particular conditions. Right. So let's begin with the first disorder, that is gene disorder. Right. So we are talking about genetic disorders. So we are talking about genetic disorders. So under genetic disorders, the first category what we are taking up under genetic disorders is the gene disorders. Right? So when we say gene disorders, what exactly does it mean? What does this actually mean? A disorder which is caused because of mutation in a gene located on a specific chromosome. And that mutation could be any of the mutations what we have already seen in the previous session. It could be either mutation, or it could be either inversion mutation, or it could be some sort of frame shift mutation, right? So all these fall under the category of gene mutation. So because of that mutation, if a specific or particular disorder is called, we start calling it as gene disorder. Now, as we have already said, gene disorders are classified into two categories, right? One is called autosomal gene disorder and the allosomal gene disorder. So there are two categories here, autosomal gene disorder, and the other one is either allosomal or X-linked gene disorder, right? Either autosomal gene disorder or X-linked gene disorder. Now, with regards to autosomal gene disorders, what did we see? Mutation in the gene located on the autosomes. Here in X-linked, there is mutation in a gene present or situated in the X chromosome, right? Autosomal gene disorder, X-linked gene disorder, right? Now, this autosomal gene disorder is again of two types. One is called a dominant disorder, and the other one is a recessive type of disorder, right? Autosomal dominant disorder, autosomal recessive disorder. So in this session, let us begin with some of the autosomal dominant disorders, right? So let's begin with a few autosomal dominant disorders. Then we'll get into autosomal recessive disorder. Then again, similarly, X-linked dominant disorders and X-linked recessive disorder. So let us take up each category and let us see a couple of examples in each of the category, right? So first, let us begin with autosomal dominant disorder. So under gene disorder, so we'll be taking up with autosomal dominant disorder. So this would be the first category here. Autosomal dominant disorder. Right. So under autosomal dominant disorder, as I've already said, what does this autosomal dominant disorder mean? There is a dominant mutation, right? Of the genes that are present in the autosomes, right? So because of a dominant mutation, right, that is located on a gene, or because of a dominant mutation in a gene that is located on the autosomes, this sort of a disorder is seen. So that condition we start calling as autosomal dominant disorder. So under this autosomal dominant disorder, let us take up with the first disorder, which is called as myotonic. It is called myotonic dystrophy. The first condition is called myotonic dystrophy, right? Simply referred to as MD, myotonic dystrophy. Now, this myotonic dystrophy is the first disorder what we'll be taking up in the list, right? And this myotonic dystrophy is an autosomal dominant disorder, right? Myotonic dystrophy is an autosomal dominant disorder. It actually belongs to a group of diseases which is called as muscular dystrophy. Muscular dystrophy. Right? So muscular dystrophy is actually a group of diseases. So one of the major disease under muscular dystrophy is this myotonic dystrophy. Now, how exactly is this myotonic dystrophy caused? Right? So myotonic dystrophy is caused because of a mutation of a gene that is located on chromosome number 90. Chromosome 90, right? On a gene located on chromosome number 90. Right? So due to mutation in the gene that is located on chromosome number 90, right? And that gene is actually responsible for synthesizing a protein which is called as myotonic dystrophy. Myotonic dystrophy protein kinase. So myotonic dystrophy protein kinase. In simple, we can call it as MDPK. So this is the protein that is actually synthesized. So in the gene that is responsible for the synthesis of this protein is getting mutated. And that gene, we start calling that gene as DMPK. So this is the gene name here, right? So this gene that is located in chromosome number 19 is getting mutated. As a result of which, what is happening? This myotonic dystrophy protein kinase is not being formed properly. Right? So this protein is actually considered as one of the predominant protein for proper functioning of the muscles. Right? So it is considered as one of the predominant protein for the proper functioning of the muscle. Now, since this protein is not formed, what would happen? Right? Since this protein is not actually formed, what exactly is happening in the body? Right? The muscle contraction and relaxation process is getting affected. The muscle is able to undergo the process of contraction, but the process of relaxation is not happening because of this protein being deficient. Right? So due to mutation in DMPK gene located on chromosome number 19, right? So the myotonic dystrophy protein kinase is not formed, which is actually considered as a predominant protein for the proper functioning of the muscle. Now, because this protein is not formed, what exactly is happening is muscle contraction is happening, but the process of relaxation is not taking place. Now, if muscles undergo contraction and relaxation, if it is not happening, so what would be the condition? Over a period of time, the muscle function will get lost. Right? So there would be loss of muscle function, and at the same time, there would be weakening of the muscles. And also there would be loss of muscle mass. As a result of which, over a period of time, the muscle contraction itself will actually not happen. Right? So that is the condition what we have, which is called as myotonic dystrophy. So what exactly is happening in myotonic dystrophy? Myotonic dystrophy is considered as an autosomal dominant disorder, right? Because of its this disorder, what are the causes of this disorder? It is because of a mutation in a gene that codes for a protein called myotonic dystrophin protein kinase, right? Which is located on chromosome number 19. And the gene here is DMPK gene. So due to mutation in this DMPK gene, which is located on chromosome number 19, what exactly is happening? This particular protein is not being formed. As a result of which, the protein function, or rather the muscle function, is being highly affected. So in these conditions, the muscles are able to undergo the process of contraction, but they are not able to relax. Now, since they are not able to relax, what would happen? Over a period of time, there is weakening of muscles happening, and at the same time, there is loss of muscle mass also happening. So because of this reason, what is happening, right? The proper muscle contraction does not happen, and the muscle itself will not undergo function, right? So this is the first condition what we have, which is called myotonic dystrophy, right? Which is considered as an autosomal dominant disorder. Right, so this is the first disorder what we have, which is called myotonic dystrophy. Right, now let's take up with the second disorder. Right, let's take up with the second disorder. Second disorder is called as Huntington's chorea. So the first disorder what we took up was myotonic dystrophy. Myotonic dystrophy, MD. The second disorder what we are going to take up is, it is called as Huntington's disorder, or we can call it as Huntington's chorea. Right, so this is the second condition what we are actually taking up, which is called Huntington's chorea, or simply we can call it as Huntington's disease or Huntington's disorder. Right. So Huntington's disorder also falls under the category of autosomal dominant gene. So what does that mean? So there is a mutation, a dominant mutation is being present, and that dominant mutation is actually present in the, or is seen in the gene that is present on one of the autosomes. So here what exactly is happening in this Huntington's chorea is, there is mutation seen in a gene that is located on chromosome number 4. Chromosome number 4. Right? So due to mutation in a gene located on chromosome number 4, what exactly is happening is an inhibitor is being synthesized. Right? An inhibitor is being synthesized because of this sort of a mutation. Now, what is an inhibitor? A substance which inhibits something. We call it as inhibitor. Right. Because of this mutation here of a gene that is located in chromosome number 4, an inhibitor is being synthesized. Now, what does this inhibitor do? The inhibitor starts acting or interfering the brain cell metabolism, right? So this inhibitor would start inhibits the brain cell metabolism. So what does this mean? Brain cell metabolism, right? Metabolism is nothing but the reactions that occur where in the brain, right? So this particular inhibitor, which is being synthesized because of a mutation in a gene located on chromosome number 4, an inhibitor is being released, and this inhibitor, right? And this inhibitor, what is it doing? It is going and inhibiting the brain cell metabolism. So if the metabolic activities in the brain was being inhibited, what would happen now? Right? There is no proper signals passed on from the brain to any other parts of the body, as a result of which it would gradually, right? It would gradually lead to brain cell damage, right? It would gradually lead to brain cell damage. So there is damage in the brain cell. Once the brain cells gets damaged, what would happen? All the other functions in the body will highly be reduced, or rather it would be inhibited also, right? So that is the condition which we call it as Huntington's chorea, right? Which is again an autosomal dominant disorder, which is caused because of a mutation in a gene present on chromosome number 4. So because of this mutation, an inhibitor is being synthesized, and this inhibitor would go and inhibit the brain cell metabolism, as a result of which the metabolic activities in the brain are being affected. Now, because of this inhibition of brain cell metabolism, over a period of time, there would be brain cell damage, as a result of which a person would lose the memory, and there would be no proper coordination between different parts of the body as well, finally leading to some sort of mental retardation, right? So this is the second disorder what we have, which is Huntington's chorea, another autosomal dominant disorder. So the first autosomal dominant disorder what we saw is myotonic dystrophy, right? This was caused because of mutation in chromosome number 19, right? So this is seen due to mutation in chromosome number 19. And here we are talking about the second disorder, which is called as Huntington's chorea, which is again because of a dominant mutation which is seen on a gene located on chromosome number 4, right? And those are the effects of this sort of a mutation, dominant mutation, right? So moving on to the third disorder what we have, right? So moving on to the third disorder. The third disorder is something called as Alzheimer's disease, right? So the third disorder is called as Alzheimer's disease. I hope some of you would have heard of this disorder. Alzheimer's disease or Alzheimer's disorder. So this is the third disorder what we have under the head autosomal dominant disorder, Alzheimer's disorder. Now this Alzheimer's disorder is considered as a neurodegenerative disorder. It is called as a neurodegenerative disorder. So what does this mean, neurodegenerative disorder, right? Neuro, something to do with the nervous system. Degenerative, that is nothing but degeneration. So there is some sort of degeneration in the nerve cells of the nervous system, right? So we call it as a neurodegenerative disorder, right? Now this Alzheimer's disease is also because of autosomal dominant mutation, right? So there is a mutation in the gene, right? There are actually two genes involved in Alzheimer's disease, right? There are two genes involved in Alzheimer's disease. One gene is located on chromosome number 19, right? And the other gene is located on chromosome number 21, right? So two genes. So mutation in both the genes, one gene located on chromosome number 90 and the second gene located on chromosome number 21 would lead to this sort of a disorder called as Alzheimer's disorder, right? So what would happen because of these mutations, right? So due to mutation of a gene located on chromosome number 19 and chromosome number 21, what exactly is happening is, right, the brain cell, again, there is some sort of inhibition in the brain cell metabolism, as a result of which the brain cells would undergo the process of degeneration, the brain cells undergo the process of degeneration. And over a period of time, all the neurons or the brain cells would be completely damaged, leading to the complete loss of memory. Right? It would lead to complete loss of memory. So they usually, the scientists say that Alzheimer's disease is an age-related disorder. So as the age of a person keeps on increasing, the chances of mutation in these two genes also would be higher. Right? So because of this sort of a mutation, right, the brain cells would be damaged, and over a period of time, due to complete degeneration of brain cells, the person will completely lose his or her memory. So therefore, we start calling it as neurodegenerative disorder. Right? So neurodegenerative disorder, we have Alzheimer's disease due to mutation in two genes. One is chromosome number 19 and chromosome number 21. Right? Again, a dominant autosomal mutation. So if you see the chromosomes here, what we have seen are all said to be autosomes. Chromosome 19 is an autosome, 4 is also an autosome, chromosome 21 is also said to be autosomes. And all of these mutations are caused because of a dominant mutations. Right? So these are the three autosomal dominant mutations what we have. Right? One is called myotonic dystrophy, huntington's chorea, as well as Alzheimer's disease. Right? So what did we see in case of myotonic dystrophy? Myotonic dystrophy is again caused due to mutation in a gene that codes for a protein called as myotonic dystrophin protein kinase, which is located on chromosome number 9. Now, because of that mutation, dominant mutation, what exactly is happening? This myotonic dystrophin protein kinase is not formed. Since this particular protein is not formed, what is happening? There is contraction of muscles happening, but there is no relaxation of muscles happening. So over a period of time, it would gradually lead to improper functioning of the muscular system, where the muscles will start becoming weakened, and also there is loss of muscle mass or muscle density happening. That is the first disorder, which is a myotonic dystrophy disorder. Second one is Huntington's chorea. Right? This is again caused because of mutation of a gene that is located on chromosome number 4. Right? So because of this mutation, what exactly is happening? A protein, or rather an inhibitor, is being synthesized, and that inhibitor, what does that inhibitor do? That inhibitor will go and start affecting the metabolic activities of the brain cell. Right? It inhibits the brain cell metabolism. It starts inhibiting the metabolic activities of the brain cell. As a result of which, the proper metabolism in the brain does not occur, due to which, over a period of time, it would lead to the damage of the brain cells, whereby a person will start losing coordination between all parts of the body, and also the memory also will start diminishing. Right? So that is the second condition, Huntington's chorea. The third condition what we saw is Alzheimer's disease. I told you that it is an age-related disorder, and also it is called as a neurodegenerative disorder. So this condition is also an autosome, because of an autosomal dominant mutation. Right? And here the mutation is seen in two genes, one gene located on chromosome number 19 and the other gene located on chromosome number 20. So due to mutation in these two genes, what exactly is happening? Again, the brain cells are not able to process properly, or they are not able to function properly, such that over a period of time, it would lead to degeneration of the brain cells or the neurons. And over a period of time, again, due to gradual degeneration of the brain cells, the memory of that person will be slowly reduced. And over a period of time, a person will completely lose his or her memory. Right? So that condition, we start calling it as Alzheimer's disease. So things what you need to remember, myotonic dystrophy is because of a mutation in gene located on chromosome 19. Huntington's disease, because of a mutation that is located, because of mutation of a gene located on chromosome number 4, and Alzheimer's disease due to mutation in two genes, one gene located on chromosome 19 and the other one located on chromosome number 21. Right? So these are the autosomal dominant disorders what we have. Right? So you need to be very clear with these disorders and under which category these disorders fall. Because in the previous session, when we were discussing about the pedigree charts, right? I told you that in your exam, the pedigree chart will be given and they'll give you 4 options: autosomal dominant, autosomal recessive, X-linked dominant, and X-linked recessive. So out of the 4, you need to select one. In some cases, what they give is, rather than mentioning which is autosomal dominant or autosomal recessive, they'll directly give these sort of disorders. So if that is the case, you need to identify these disorders will fall under which category, and then you need to interpret the disease and the pedigree, right? So make a note of all of these disorders, what we are actually taking up, right? So this is what we have with autosomal dominant disorder. Now let's take up with autosomal recessive disorder, right? Let us take up a few autosomal recessive disorders and we'll see how are they caused and what do they do. The second category of disorders what we have is the autosomal recessive disorders. The autosomal recessive disorder. Now, unlike autosomal dominant disorder, we said that the mutation is said to be a dominant mutation in the autosomes, right? So when we say dominant mutation, the condition has to be both homozygous dominant or it could be heterozygous dominant. If either this is the case or this is the case, the disease would be expressed. But in case of recessive disorders, right, the condition should definitely be this one. Only if this is the case, right, only if this is the case, this sort of recessive disorders would be expressed, right? Till now, what we saw autosomal dominant disorder is either it was this or this mutation, right? But now when we talk about recessive disorders, right, so the recessive condition is responsible for the expression of that particular disorder, right? So these are what we call it as autosomal recessive disorder. Under autosomal recessive disorder, let us take up a few diseases and we'll see how are they actually caused. So under autosomal recessive disorder, the first disorder what we'll be taking up is what we have already discussed in the previous session, right? The first one is called as Alkaptonuria. So I thought we'll just go through it once again, right? So Alkaptonuria is actually the first metabolic disorder that has been identified by Sir Archibald Gary, right? So it is usually abbreviated as AKU, right? So you need to remember the abbreviations also, because most of the cases, rather than full form, the abbreviated forms would be given. So Alkaptonuria is usually abbreviated as AKU. This is considered as the first inborn metabolic disorder which was identified by Sir Archibald Gary. Right, so we saw a few things with regards to this yesterday. Right, now we said that Alkaptonuria is an autosomal recessive disorder. So what does that mean? If this disease has to be expressed, a recessive mutation is required. Right, if this disease has to be expressed, a recessive mutation is required, and that mutation is caused due to mutation, recessive mutation in a gene located on chromosome number 4. Sorry, chromosome number 3, right? The gene located on chromosome number 3, right? And what is that gene, right? I told you that in the previous session. In normal human individuals, what exactly is happening is a highly toxic substance or a compound called homogentisic acid. Homogentisic acid is being formed. Right, in normal human individuals, right? In normal human individuals, a toxic compound called homogentisic acid is formed. Now, this homogentisic acid, remember, we are talking about in normal individuals, right? Now, this homogentisic acid, which is formed in the body, goes to the liver, and in the liver, there is a process called as detoxification. Right? We know that one of the major functions of the liver is undergoing a process called as detoxification. So whatever toxic substances are formed in the body, they are actually converted into non-toxic or rather less toxic form before it is eliminated out of the body. Right? So here also, similarly, we have a toxic compound called homogentisic acid, which is actually being synthesized in the body. Right. Now, this homogentisic acid will go to the liver, right? In the liver, they undergo a process called as detoxification. And due to this detoxification, homogentisic acid is getting converted into a less toxic form. Right? Homogentisic acid is getting converted into a less toxic form. So for this conversion, an enzyme is required, and that enzyme here is called as homogentisase. Right? So this enzyme called homogentisase is involved in the conversion of homogentisic acid into less toxic form in the liver. So this is the normal process. Yesterday I told you that homogentisase is also called as, there is one more name given for it. It is also called as Alkapton oxidase. Right? It is also called as Alkapton oxidase. So this is the enzyme which is responsible for the process of detoxification of homogentisic acid in the liver. But in case of this condition called Alkaptonuria, what exactly is happening? This condition is caused because of a recessive mutation in a particular gene located on chromosome number 3. And that gene is the gene which codes for an enzyme called homogentisase. Right? So due to a recessive mutation, right? So due to a recessive mutation in a gene coding for an enzyme called homogentisase or Alkaptonuria, which is located on chromosome number 3, this sort of a disorder is seen. Now what happens if there is mutation in this particular gene? Now because of mutation in this gene, this particular enzyme is not being produced. Now, since this enzyme is not being produced, what would be the condition now? Homogentisic acid is not getting detoxified, right? So there is no process of detoxification happening. Now, if there is no process of detoxification happening, what actually would occur now? Because of no detoxification, the homogentisic acid concentration in the body would start increasing. So there is increased concentration of homogentisic acid in the body. Now, once the concentration of homogentisic acid in the body will start increasing, it gets accumulated in various parts of the body. So where all does it get accumulated? It gets accumulated in the cartilage, right? It gets accumulated in the cartilage. Then it gets accumulated in the joint capsules. Then it gets accumulated in the ligaments. It gets accumulated in the ligaments. Then it would get accumulated in the tendons as well. So these are the places where this homogentisic acid would get accumulated. So what exactly is happening? Due to a mutation in the gene coding for an enzyme called homogentisase, which is located on chromosome number 3, right? The toxic homogentisic acid does not undergo the process of detoxification, where it is not getting converted into less toxic form. Now, since it is not getting detoxified, what would happen? The homogentisic acid concentration in the body will start increasing. Right. Now, because of that increase in the concentration of homogentisic acid, it would start getting accumulated in places like the cartilage, the joint capsules, the ligaments, as well as tendons. Now, once it gets accumulated in all of these places, what do you think would happen? Right? The process of movement is actually being affected. So, since it gets accumulated in the joint capsules, we will not be able to move our particular bones, right, which are actually connected to different bones. Similarly, the ligaments and the tendons also. We know that they are actually connect the bone to the muscle and the muscle to the bone. The normal process of movement in the body would be affected because of the accumulation of homogentisic acid in all of these areas. Right. Next one. What exactly is happening? Now, as the concentration of homogentisic acid in the body will start increasing, some of this compound would be excreted through the urine, right? So some of these compound, that is homogentisic acid, is excreted through the urine, right? Now this urine containing homogentisic acid, if it is exposed to air for some time, right? If it gets exposed, or if it comes in contact with air for some time, right? It would turn black in color. right? It would turn black in color. And this black color of urine is because of the oxidation of homogentic acid in the presence of air, right? So in the presence of air, homogentic acid will start undergoing the process of oxidation, as a result of which within a very less time, right, the urine would be turned black in color. So that's the reason why Alkaptonuria is also called as black urine disease, right? It is also referred to as black urine disease or black urine disorder, right? So this is what we have with Alkaptonuria, which is considered as an autosomal recessive mutation caused due to mutation in the gene coding for homogentisase enzyme, which is located on chromosome number 3. Right? So this is the first autosomal recessive disorder, and along with that, this was the first disorder, genetic disorder, inborn genetic disorder to be identified. Right? So now let's take up with the second type of disorder, second type of autosomal recessive disorder. Right? So second type of autosomal recessive disorder, what we're going to take up is also a metabolic disorder. Right? It is also a metabolic disorder, which is called as phenyl ketonuria. Phenyl ketonuria, which is abbreviated as PKU. So Alkaptonuria, AKU, and phenyl ketonuria, PK, phenyl ketonuria, right? So this is also a metabolic disorder, just like Alkaptonuria, metabolic disorder, right? And this also is said to be an inborn disorder, right? It is also considered as an inborn metabolic disorder. Right? So in Alkaptonuria, there was mutation in the gene located on chromosome number 3. Whereas in case of phenyl ketonuria is caused because of a mutation in a gene which is located on chromosome number 12. Right? So due to mutation in a gene located on chromosome number 12, this sort of an inborn metabolic disorder called phenyl ketonuria has been caused. So if you could recollect, sometime back we were actually talking about this sort of a disorder, phenyl ketonuria, when we were talking about pleiotropy during the process of inheritance patterns. When we were talking about pleiotropy, we came across this sort of a disorder, right? Okay, so the same thing what we are going to discuss, phenyl ketonuria. So what exactly happens during this condition called phenyl ketonuria? Before getting into this disorder, let us see what exactly happens in our body in normal condition. Right. In normal condition, we have an amino acid called phenylalanine. Phenylalanine is an amino acid which is actually considered as an essential amino acid. So when we say essential amino acid, what does that mean? The amino acid which is not synthesized in our body, but we need to take it through the external diet. Right? So this is considered as an essential amino acid. Now this phenylalanine in our body, right, would be converted into tyrosine. It would be converted into an amino acid, another amino acid called tyrosine, in the presence of an enzyme called phenylalanine hydroxylase. So in simple, we call it as PAH enzyme. Right? So in the presence of this enzyme called PAH or phenylalanine hydroxylase, the phenylalanine amino acid is being transformed into another amino acid which is called as tyrosine. Right? Phenylalanine getting converted to tyrosine. Now what is the role of this tyrosine? Tyrosine amino acid will start acting as a major precursor for the synthesis of neurotransmitters. Right? For neurotransmitters such as, we have a list here, such as we have something called as dopamine, which is one of the neurotransmitter. Then we have epinephrine, which is also a neurotransmitter. I think you would have studied about epinephrine. And then there is again norepinephrine also, which is also a neurotransmitter. Epinephrine. So for synthesis of these neurotransmitters such as dopamine, epinephrine, and norepinephrine, right? So this amino acid called tyrosine plays a very, very important role. So it would start acting as a precursor for the synthesis of these neurotransmitters. Now, what is the role of neurotransmitters? We are very much sure about that. What is the function of this neurotransmitter? They are responsible for the transmission of impulse from one neuron to the other neuron in case of a chemical synapse. So without the neurotransmitter, there is no impulse transmission happening from one neuron to the other. So these neurotransmitters play a very important role. For the synthesis of these neurotransmitters, this amino acid called tyrosine is actually required. So without tyrosine, these neurotransmitters are not included. So in normal normal condition, this is the normal mechanism that is happening, where the phenylalanine amino acid is being transformed into amino acid called tyrosine in the presence of enzyme called phenylalanine hydroxylase. Once the tyrosine is being formed, this tyrosine will now start acting as a precursor for the synthesis of neurotransmitters such as dopamine, epinephrine, as well as norepinephrine. Now again coming back to our disease condition here, phenyl ketonuria. So what happens in phenyl ketonuria? As we have said, phenyl ketonuria is a metabolic disorder caused due to a mutation in a gene located on chromosome number 12. So which is that gene? That gene is this one, phenylalanine hydroxylase gene. So due to hydroxylase gene, which is located on chromosome number 12, what is happening here? This enzyme is actually not being produced, right? So due to mutation in phenylalanine hydroxylase gene, which is located on chromosome number 12, right? So this gene is not being formed. So if this gene is not formed, what would happen? The entire metabolic processes would be affected. Tyrosine is not formed. Since tyrosine is not formed, the neurotransmitters are also not formed. So what would happen? Two conditions will be seen as a result of this. First condition, since phenylalanine is not getting converted to tyrosine, what would happen? Phenylalanine concentration in the body will start increasing. Now what happens if phenylalanine concentration in the body increases? This phenylalanine, whatever is there, with increased concentration, would be transformed into another compound which is called as phenyl pyruvic acid. It would get transformed into phenyl pyruvic acid. Now this phenyl pyruvic acid will migrate to the brain. It would migrate to the brain. Once it goes to the brain, what would happen? This phenyl pyruvic acid will start destroying the brain cells, or it would lead to degeneration. It would lead to degeneration of brain cells. So this is the condition seen because of accumulation of phenylalanine in the body. Get it? So this is the first effect that is seen because phenylalanine is not getting transformed into tyrosine. The second condition that we see here. Now, since phenylalanine is not getting converted to tyrosine, right? Tyrosine is not available for the synthesis of neurotransmitters. Therefore, these neurotransmitters such as dopamine, epinephrine, and norepinephrine, they are not synthesized. If they are not synthesized, what would happen now? There is no impulse transmission happening across the neurons. If there is no impulse transmission happening, there is no coordination between different parts of the body. As a result of which, the individual here will start suffering from something called as mental retardation. One side, the brain cells are getting denatured or degraded, right? The second one, what is happening? Due to the improper formation of neurotransmitters, what exactly is happening here? There is no proper coordination happening within the individual, right? So two effects are seen because of this condition called as phenyl ketonuria. First one, what exactly is this phenyl ketonuria? It is actually caused due to a mutation in the gene, right, which codes for an enzyme called phenylalanine hydroxylase located on chromosome number 12. Now, because of this mutation, phenylalanine is not getting converted to tyrosine, right? As a result of which, the neurotransmitters are not being formed. So, if phenylalanine is not getting converted to tyrosine, what would happen? The phenylalanine concentration in the body will start increasing. Now, because the phenylalanine concentration in the body increases, this phenylalanine, which increased concentration, will now be transformed into another compound called phenylpyruvic acid. Now, this phenylpyruvic acid will migrate to the brain, and in the brain, it would start degrading or degenerating the brain cell. Once the brain cells would get degenerated, what would happen? The mental stability of that individual is lost. Therefore, a person would be mentally retarded. Second condition, what we have seen, because phenylalanine is not getting converted to tyrosine, right? So tyrosine is not available for the synthesis of neurotransmitters, right? The neurotransmitters such as dopamine, epinephrine, and norepinephrine are not being synthesized. As a result of which, there is no impulse transmission happening between the neurons. Since there is no impulse transmission happening, what would be the condition here? Right? There is no proper coordination happening within the system. So these two are the things which are actually caused because of this condition called phenyl ketonuria. Right? It is again a metabolic disorder because the proper metabolic activities are being affected here, and we also call it as an inborn disorder. So by birth, the child usually the child has this sort of a mutation, right? So two disorders we have seen here in this session with regards to autosomal recessive disorder. Three disorders we have seen with regards to autosomal dominant disorder. One was called myotonic dystrophy. Second one was Huntington's disease or Huntington's chorea, and the third one was Alzheimer's disease. With regards to autosomal recessive disorder, we have Alkaptonuria as well as phenyl ketonuria. Right? So we'll stop here for today. In the next session, we'll take up with some more disorders pertaining to autosomal recessive disorders. Then we'll move into X-linked dominant as well as the X-linked recessive disorders. Right? Thank you.